Just a second, guys. Just a second.
Okay.字幕，字幕，开启字幕。
Am I audible, brother? Hello?
Hello.
Am I audible?
Yes, I can
Sure.
hear.
Can anyone share his screen and I can see like what he did?
Okay, okay.
I'm waiting for you. Share your screen.
Screen. List. Can you watch my screen?
Wayne, just share it to me. Share it to me to online. I'll check it. Both of you just share it to me. The file. It's expired online, so
Okay, wait a second.
Let me share. It's visible now. So have you guys selected any gene out of this? Any gene that you like are interesting on?
Okay. Well, we just pick some. So you mean the genes that we filter out that any we interested to research?
Mm-hmm.
We're not pick it
Look.
up.
Wait. Before selecting like any gene, just like I guess I have told you about gene card, like to find out the family members, like how many family members are there in this. Last time, did I tell you about the gene card?
No. No?
Okay, no problem. Wait,
Okay.
let me give you the link. Okay,
Okay.
before that, like we missed 2 3 meetings. So before that, let me just start you with some basic like what we are going to do in bioinformatics in future. So
Okay.
I have this. So these are basically the material and methods that we used to say these are material and methods. Okay, basic methods like these are just basic method. I'll just share it to you. Just note it down in your notebook or anywhere where you're just writing the reports. Okay. So I'll show you first. I'll show you how all these plots and graphs look like. So the GPR database. So you guys, I guess, have seen already the GPR. We
Yes.
have work on GPR like last time. Mm, have a lots of so GPR is this. So what we can do on GPR, we can just take out the data from GPR and we can search whether it is cancerous or non-cancerous, significant or non-significant. Okay. What else?
Okay.
What else we can do here is like we can check the survival analysis over here also. I will tell you later on like what survival analysis is. Like you can find that the prognosis of the cancer gene is good or not, the number of days that the patient survived. You can see all this from the survival map. So we have 2 3 data sets from where we can take out the survival map. uh, like I have mentioned km plot. Okay, this also you can like take out the survival map from km plot from GPR, and we have TCGA, the Chinese Globe Atlas. Wait, I'll tell you the full form. TCGA. So the Cancer Genome Atlas. Okay, TCGA. From here, you guys can also take out the survival map. So GPI you have covered. KM plot is used for taking out the you can say the survival map. Okay, then Venn diagram. Venn diagram we is like Venn diagram, then Ulkan database, volcanic plot, heat map. This overall survival means like the number of patient and how what is the chances of their survival. It's a kind of a plot, just like a KM plot, survival map and overall survival. Correlation plot, like when you will work later on, then when you will go for protein interaction. Okay, so the gene will interacting let me show if I have the results. I'll show you the results. Okay, so maybe you can get better. Overall volcanic plot. It's not complete. Wait. GPM. If you guys don't understand anything, just do let me know in between, so I can just clear your doubts also, or just write down your doubts and just send it to me. I'll send it to you online. Desktop GBM. Report. So I just send you the material and methods. Okay. So what you have to do, you also when you'll find your gene. So in the end, you guys have to find some key points about your gene. From the gene card, you will get the full name of your gene. Like when I'm working on GBM, okay, it's a brain cancer, fourth stage. So you will get your full name of this, and other you have to read some papers regarding like the gene that you're working on. You will get some papers regarding not like bioinformatics, but some wet lab work. So you have to see like what's your gene specific role, what it helps, like if it's down regulated, up regulated, like its prognosis is poor or not. So this is a few information regarding my gene. Then you have to take out the basic gene family information. I'll tell you about the gene card from where you can just take out the gene family members. Like I have selected this gene. So on gene card, when I just put PTG3, so there are many genes list, but you have to just find out your family members. These are the family members of this gene. Then you have to check whether they are cancerous or significant. These things you will get to doing later on, like when you're going to submit a paper, when you need some additional details. So this, what is this GPA, from where we find that it is cancerous and the star says it's significant. Then this is a survival map. Okay. And this is the pan cancer. This we have taken out from this material methods. Where's GPM? Yeah, from Ulken database. This is at the website, our database, from where we can just take out these kind of information. Just a second. This like it's just showing you the my number of. Look, it's in GBM. This is the pan cancer, means all the cancers. Okay, so it's all the cancers: breast cancer, my GBM, lung cancer, your pancreatic cancer. So it's just telling you that the number of normal cells and the number of tumor cells. The red one indicates the tumor cells, and the blue one indicates the normal cells. So it's just telling you the relation between these two genes, sorry, and that the tumor number of tumor expression is higher than the normal one in the in the GBM. So you also need to check out the pan cancer analysis. This is a survival map, the overall survival and the recurrent survival. Okay, so what recurrent, here you will ask you common question, that what is recurrent glioma? Okay, anyone know what is recurrent glioma? Recurrent means
Means you've been treated by the cancer is uh,
again the chances of coming it right after
yeah.
treatment. Yeah, you're right. Like again, what's the chances of that cancer cell will grow again after that treatment? So this is
Yeah,
the
yeah.
detail of that, and this is the overall survival from the glioma. Okay, later on when we will do
Okay.
this, I'll explain you what this red and what is this blue indicates. Today I'm just giving you the small information, okay, regarding what's how it look like, so you have all the details. So this is the Go enrichment plot. I will explain you later how it, like what it indicates, what's KEGG pathway, what's enrichments. Okay. Then this is the volcanic plot. We'll take out some data from the CBio portal. We have also here CBio portal here I did CBio portal. We have will this portal will help you to take out many like data for RNA expression, for heat map, for volcanic or correlation plot. We'll take all the RNA data from the CBio portal. And this is from the HPA, the Human Atlas. This one, it's the Human Protein Atlas (HPA). When you will visit the site, you will get to see the tissues, the normal tissues, and the infected tissues from cancer. Look like this. This is a normal tissue where we'll get this is the glioma tissue present in cerebral cortex. This is the normal, and this is the glioma tissue. Okay, and this one is a little high. This is then the timer. In the timer you will get to see the immune infiltration. Okay, so this will indicate about the purity of the cell, the T cell CD8 plus timer CD4 neutrophil macrophage dendritic cells and B cells. I'll just let you know slowly how you can read this graph, what this line and what this dot indicates. When you will, then this is the protein interaction. When you will finalize your gene, so you will just put like the family members of a gene. These are the four family members of my gene. So when you put in the website database, so it will tell you that these genes are interacting with other genes. These color nodes and these lines indicate something. You will get to know, you will read this from the website, that what these lines and what these nodes are indicating. Like my gene is BTG3. Okay, I'm working on this gene, so this gene is not directly interacting with their family members. Okay, and let me just show you an example, live example here. Protein string. So this is the website from where we'll get out our genes, multiple genes. I'm just working today. I'm working on the lung cancer gene. So this is my lung cancer. I have three family members: MTX1, MTX2, and MTX3. So luckily these three are interacting with each other, but for my GBM cancer, they are not interacting directly with each other. They are like, this is BTG2. So they are interacting with each other with the help of C08 and C07. Like, they're not interacting here directly, not with BTG4 even, and this. If you see, BTG3 is interacting with C08 and then interacting here with BTG1. So, but in this case, you can see they are interacting direct with each other. Look, 1 2 3. So these notes and these color indicate something. So here when you will take out the gene, you can see that what these color nodes are indicating, what these color nodes are indicating. You can just read all the details over here. And after that, this is how protein interaction looks like. Okay, if your gene is interacting with each other directly, just like this one is interacting directly, so you can just take out the correlation between them. Correlation means like what activities, what p values, what adjacent value or correlation value are similar with each other. In the correlation plot, uh, this is the correlation plot. Like, I'm just taking out the relation between BTG 1 2 3 4. Look at the numbers. It's in minus zero point 0.06, zero point 0 minus. Why? Because they're not interacting with each other directly. But when you see here in MTX correlation, but my lung cancer was interacting with each other directly. So look at the numbers over here in this correlation plot. 0.3228 minus 0.32 minus 28. So look at these number, they are just stronger. And the p value, this color indicates the p value. Look at the p value, it's strong. The red one, 0.8, 0.4, somewhere 0.2. And after this correlation plot, then we will go for the mutation plot. This is also came from the CBio portal. and this is the heat map. The heat map of these data are from the timer. This is the immune infiltration data from the, this is a heat map that indicating, like the P indicates for P value, and the RHO indicate for the significant of the gene, like how significant it is in your cancer. Then this is the GSEA. This is we will do to find out the pathways in the end, and this is the meta code that we used for finding the pathways. So I'm just currently also doing the meta core for my lung cancer today. So the so when you will come to the meta core, like we will do this using the server of TMU. Okay, so when you take out your gene from the CBio portal, you need to upload the data over here, and then you should go for the show location for the gene. After uploading the data, you will just get to show location of your gene. Then you will see this 50 kinds of pathways for your gene. Yeah, for that gene that you're selecting. So you have to just not downloading all the 15 pathway. So when you will complete this process, GSEA. So you will get to see some common pathways of GSEA and Metacore. So you have just have to download those pathways from the Metacore, not all the pathways. So how it look like? It's like look like this. Okay, I'm just it's still it's under process. Like it's just the process. The image is still processing. So after that, I'll just let you know what these things are. These things means. You will get to download a legend from here, and legends tell you that what this symbol is genetic kinesis. This symbol is protein kinesis is happening in that thing. If this is happening in that between this and that protein, the protein are binding between two genes or two the pathways, and this is this indicate the genetic receptors. So this will tell you that like what process are going on here, over here, here. Okay, so now what you guys have to do for this week is to find out the can anyone like share me all the gene list, like you have total number of gene list also. Both of you just share me
Mhm.
that list, like these are just cancers and significant. I need the all the list. Okay, what if after like if you find this gene as suitable for your research, so you should go on the gene card. I'll share you the link of this gene card, and you have to paste your gene over here. Where is this? No results. I guess I need to review because I'm not getting any gene name by this gene symbol. Registration values. Just send me the whole gene list, any of you. One whole gene list that you downloaded.
Oh, shit.
Because I don't know, gene doesn't look like this. It's a different symbol numbers. Yeah, it should look like this. You should choose cancerous significant. Yeah, gene should be look like this. If you are getting any gene name by zero point 0.6 or any dash in this, just remove those gene. Don't use this.
Okay.
Don't use this. Use this one like GFRA1, SCUB2, LY6. Okay. So just use
Okay.
this one. Just delete all this dash 1.1. You can use this one. Just remove this. Remove
But
this
what
from
about
your
the
list.
p-value? Because the green mark is like the p-value is over 0.05%.
Just a second. Let me see this. we can neglect in this one because this gene will not uh, work in your research.
Okay.
Like if it's point, let's like if it's not 0.05, so you can just take 0.04 as that, and we will mention in the paper that it's like 0.04. It's not big,
Okay.
like, so we will do in the, like, when we will do further research, just remember that the p value was this much. So we will put in that also, because we need to put all the p values in different plots, that we
Yeah.
need to use p value less than this. So my ideal was 0.05, like everyone is taking the ideal 0.05.
Okay.
Battery, one
Okay.
second, let me just charge. But just remove this kind of name, RP44, and because this will not work when you will put this in any plot. Just like I put in on the gene card and it says the gene name is invalid. But look, this is cancerous and significant. If you put this one on gene card, so it will show you the gene family name. And don't use RNA genes, only use protein coding. And also, like, when you will export this, I will tell you the shortcut how to find the family members. Export to the Excel, put this name, export to the Excel, open the Excel, and just remove all this. It's not for your use. Just simply find GFRA. You will get only, not here, here. Just put the filter, search GFRA. You'll get this much GFRA. So you just need to use GFRA 1, 2, 3, 4. Don't, no need to use GFRAL. So delete this INC. Just delete this. No need to use INC one also. And PDGFRA. No need to use this also. So you have four family members of this GFRA 1, 2, 3, and 4. Okay. So just take it out. Make a column over here. After this here, just write gene symbol, family member, and put GNFRA, just like put GNFRA over here, something, and in this four column, put the family member name, like 1, 2, 3, 4. 1, 2, 3, 4. Okay, put the four one, and then just go for the second gene. How many family members they have, just put that. After that, when you put out the family members, then check for the cancerous or non-cancerous and significant and on in this column, whether their family member is cancerous significant. And after that, the choose the least number of family member, like it's a four family member, and some of them are cancerous and significant, so use this one for your research. Okay.
Okay. So we still need to filter out uh, the
Family
p-value
member.
and okay.
No, not any, no any, not any value like log 2 FC value or adjacent P value. Just you need to take out the family members, and after that, yeah, and just take care of this. No RNAs. Like if you see this INC one, this is RNA gene. You need to go for this.
Yeah.
And when
I
you
mean,
open
what
this,
we going to search the names is
hmm,
from the last week we do the assignment and
hmm,
just search every genes or we need to search what we filter out.
I will just tell you one more shortcut, okay? Like what I did is like I just first I take out all the cancer and non. Wait, I took out all the cancer and non-cancers like you guys. Okay, then
Yeah.
then I found the family members. Then I go for like to see whether this gene has publications or not. But I will suggest you
Yeah.
what the shortcut for this is. Like this is just for your fast work and shortcut. Just see this, copy, paste to PubMed, go and see if it have publications or not. If have zero publication, then find his family member.
Yeah,
Okay,
but every green mark is zero publication. But without the green marks is all have been published.
because look, this gene is not you cannot use this gene for research because it's don't have any significant name or anything. That's why it's a
Yeah.
publication. So you have to
Okay.
just take out this thing. Like you will get some any of the. Just don't go for p-value right now. Okay? Just go for the cancers and significant for now. Okay, if it's significant,
Okay.
then we will adjust the p-value later on. Okay, first now just go for if it's cancers and significant, just go for the publication. Just check for the publication if it have or not.
Okay. So now
Like we
just
have a
we have
lot of
to
gene.
check the publication.
Yeah, if it's not coming in top 200, then you have 1,000 gene. Then go for next 200 genes and just check in that
Okay.
So we
because
check. So we check top 400 gene or
200.
top 200.
First check top 200. If it's not coming in top 200, then go for other 100. It's again not coming in other 100, then go for next 100. Maybe it will come
Oh,
somewhere na one gene. After that, after the gene process, it will very easy to do all this process because all this data you will get on the website of different website. You just need to download, rearrange the data, and you have to just put these things. Like I'll show you an example. Like after I have BTG, so easily I just put the data. I got that protein interaction. The same, I will go on CBio portal. I will just put the name of my genes, and in the end I will get the data for RNA expression, and then I have to put that on the. There's another website for correlation plot. I just have to put the same data over there, and it will give me the correlation plot. Same like others, just need to finalize the gene. It will take time. For me, it will take 2 months to take out the gene. And after that, it was just for 1 month completing all the processes.
okay.
We have a lot of like, just note down this. The main thing is this, that you should know what things like we have to do on later on. These are the main things that we have to do. This plot you will get automatically on the website. This plot you will also get, this plot, HPA you will also get. Like, let me show you. We have selected a gene, GF something. Let me see that. GFRA1, okay. So we will go for HPA. I'll show you how HPA and timer works. HPA and timer. So you just need to enter the gene, search it. This is the gene's full name. The tissue, the brain tissue will go in the tissue. Internet. I guess the server is down of the site. Okay, this is a tissue. Okay, you will just need to. What was the cancer of this GNA? Wayne, which cancer you are working on? Hello, Wayne. What cancer you are working on?
Breast cancer.
Breast cancer. So
Yeah.
Me is
here,
stomach.
yeah, same. You will get all this data over here. Where is the website? Here. So you are working for breast cancer. For stomach, we will
Yeah.
see if we have stomach here. Stomach cancer. For but this gene is for breast cancer. So we'll open the breast cancer, and here you will get the data of the 69-year-old female. So these are the genes. Look. So this is the female gene, the tissue. The age is 75. This is the this situation at duct carcinoma. Look at this. Cancer. This is a cancerous gene. Look at here. Look.
Okay.
So you will get to see all this data automatically when you'll find out the gene. Okay, we have different age, like a female age of 93. This is the female age of 51. How the tissue looks like. Same in the timer. That immune infiltration, you will just need to put the name of your gene, and we have to submit that what immune infiltration you want to do, like in T cells CD8 plus. Then it will just tell you the data. This website not work every time. Submit. Okay. From here, then breast cancer is BRCA. So you will see here. So this is immune infiltration, and the purity is like the, so just see the graph. It's in the positive way. RHO is a significant and P is the P value. The significance is 0.128 and the P value is 0.04 something.
Okay.
Okay, so just you need to just take out the gene finally, and data will be just, if you know how to take out the data, you can just take out the data within a week or within 2 weeks. After you will take out the data, I'll just let you know how to. But you guys need to just read some papers. Like when you go to PubMed, might just go and search for some breast cancer bioinformatics analysis. Okay, so there will be some papers for breast cancer bioinformatics. So you need to read these papers, and just you also need to tell me like what extra they have done. Like I don't know what full, like just a second. They use this Zena browser for taking out the data. I'll let you know also how to use Zena browser. Uh, the difference is that Zena browser is also taking data from different server, and the GPA is also taking from the different server. Results. So like this is from the Ulkan. I'll let you know how you can see this from the Ulkan. Okay, this is the just the difference between the normal, the blue one is the normal, and the red one is a tumor cells. And this is also from the Ulkan database, the table. This is a KM plot. You can see this KM plot, overall survival, relapse free survival, and this is the heat map. They use heat map. So the correlation plot, you can see the correlation plot. They're just showing the correlation between each other. Then again, GPA, the gene is significant and cancerous. This is the survival map, the prognosis, and next what they use. Like they just use this much information. So in the short information also we can publish a paper, or we like just the GSA map. They don't go for any Metacoro or anything. So just need to take out, just target a paper like how they are using. So just go for 2 3 papers. We need to see like what data they're using, so we can just see. For my paper, I'm just taking out some data. They are using some mutation bars, they are using some GSEA, they're using IP also. It's also a pathway analysis, some pathway analysis. So just go for some paper for breast cancer and stomach cancer, and we'll see the scatter. They have the, like they use scatter map. This is the scatter plot, and then this is the heat map. Again, the GPR. This is the enrichment, geo enrichment. So this is from a different website. We can also use this also for the paper geo. This is also showing that cell cycle, cellular DNA application, how p value, the color showing the p value, p adjacent value, and these are the pathways that. So any questions for now? Like when next maybe this Sunday or Monday we can have a meeting and we can finalize the gene, so we can go for next further like what we can go for these processes, all these things. When you guys are coming over here, any fixed date? Date is
Uh,
fixed for coming here.
like
We decide uh, July, so
like maybe
July 4th.
4 or 5
July is
July.
next week. Next
Yeah,
week is July.
yeah.
Mm,
Any 4, 5 Monday?
Like Tuesday or Thursday, Tuesday or Wednesday.
Next week.
yes.
Yeah, next week.
Yeah, next
Okay.
week.
Yeah, then great. We can directly do your research over from here. So
Okay.
maybe on Sunday, I guess on Sunday, if your guys are free, so we can just see like what you have done, the finalized gene. So when you will come here, so you can show professor that you have a finalized gene and you have done some work and you have a list of work that this work I have to do in the future, and prepare with a paper like this is a paper and I just read this paper and I need to mimic this paper for my research work, or I just need to like do these results only for my paper.
Okay.
And when you will read the paper, you will get to know that if you can increase the p value or just you have to put 0.05 p value. Anything else?
It's all good.
Okay. So you guys are coming to work on cancer cell lines or any others? So you know how to do like cell culture?
Yes.
Yes.
We've been practicing for like 2 months.
Oh, great.
Okay.
Yeah.
Great. And
2 months.
I will learn. I learn this cell culture from you guys. I'm still learning the cell culture.
Oh,
I am
Oh,
good at
okay.
bacteria culture. I can do. I'm just doing bacteria culture from last. In my whole bachelor's, I did bacteria culture. So if you need any help for bacteria, for any microbiota, I can help you. But cell line is first time for me. Still learning the cell
Okay.
lines.
Okay.
Okay, see you then Sunday or maybe
okay.
directly on Wednesday.
Okay.
Okay,
Sunday. Okay.
Thank you.
Thank
okay,
you.
okay.
Okay. Goodbye.哦，
好好笑。
这次有听得比较懂吗？
我总觉得因为不上啥，我筛选出来那些绿色的是，他都说我那些绿色不行，那我什么可以？我就问，
那他是没有看我的，他是不是都在？他就
他
说我
是拿我的示
们都不行。
范啊，
对对对，
他是
那为什么？
说筛选出来的那个就是都没有资料，那些都不
对，
是啊，看起来都不像
他是说
基因，
p
为
value
什
吗？
么啊？
他是说
就是
p
我
value 的问题吗？
说我们那时候
还是
设0.05，然后下去
对
看，然后筛选出来那些都没有，都不是吗？
对
然后他说，那你可以就是什么把 p value 降到0.04什么之类的，
04，
就往下降啊，意思是我们要全部重做一遍哦。
可是为什么这样？我就怪。对，我刚刚可以，
不知道，
因为他原本不是还说就是可以到下一个200，对。然后我问的时候，他就说就是一样，就是 top 200，所以就是我们一样重新用的意思咯。
应该吧？干做白工哦，fuck！
所以就是我们先，所以我们就是我们把，
好好笑，他把我的 fuck，
他会消音，他知道是脏话会消音，
确实，
所以就是我们p value 降到0.04，然后再重新找 top 前200个，
应该是吧？他说我找那些看起来都不像基因呢，讨厌。
对啊，可是为什么？然后，
不知道，
就是他是不是有举那个 GFRA1？
哦，
对，他是说像这种的还是基因是吗？GFR 1 2 3 4，
是吧？
不是、啊，是你的鸡都长得太丑了，
啊，
是你的鸡那个名字都长得太丑了。
你看呢、
我知道啊，
就
我
对啊，
我知道，那时候我看到啊，我的是我看一下哦，
我的只要有标绿色都是没有文献的啊，所以
对，
它又没有文献，那这样子就只能往下
都是那些
面去找，因为这
很丑的。
前面都对啊都被做过了，怎么可能那
他
个？
刚刚是不是有用你的找一个，然后呢，就是那个 data 是零，是吗？
就是因为我有筛
用
选
Gene
p
Card 去
value 嘛，
查嘛，对不对？
我看一下 p value 0.05，然后是
小于
这样子吧？
这个，嗯，
诶，
是 leg o 吗？
不知道，我不知道他怎
也
么弄的，
是那个应该是没差吧？地震地减应该是没差吧？
对吧？诶，他为什么一开始会是只有
嗯，
cancers significant？诶，那为什么会这样？等一下，重新看一下。
嘿，
筛了1、2，诶，为什么是这个？对啊，这边筛 p value，然后 cancer significant。
嗯，对，你的这张长好丑。啊，我刚刚是有随便去找一个
cell
基因，
color，
然后去基因卡，就是用那个基因卡那个网站去查，然后是有找到东西的，
哦，
就是
我看一下哦，这边现在是有大于0.05的吗？诶 less than，
这个
它
类对是
是说是
要
less
小
than
于
还是
对
greater？
小于
小
less
于
than，
小于零点零
嗯，
五是 significant 嘛，对不对？
对对
然后要是 cancerous，然后跟 significant，
significant，
就这些啊。哎，啊，我的绿
嗯，
色怎么不见了？为什么？
被
看一下
吃了。
cell color，还是把这个取消
哦，
掉？
看一下
怎么取消？
我这边，
哎，怎么取消啊？还是可以哦，这样子，这样子。对啊，就是从这边去找吧。啊，这边有绿色的，就是没有文献的啊。
对，
对啊，还是这个，对啊，这个应该也是都有文献了吧？看一下 PubMed，
啊，
开始
应该是
PubMed。
有。
对啊，有文
哎，
献啊，
你只会这
啊，这
样查？
种才没有文献吧？
因为我那个时候是还
啊，
有还有还有打那个，就是像你的话就要打 breast cancer。我的话我还会这样打，因为
哦，真的
我后来
吗？
发现，对，我后来发现你单纯打一个基因，然后跟你打这个基因再加上这个癌症的话会不一样，因为可能我也不知道，可能这个基因还有涉及到其他癌症。它、如果是单纯像我那时候就是打就是这个基因，然后加 stomach adenoma carcinoma
他示范的
这样子全部
示范的时候，
打出来。
他有说要打哦，
他示范的时候是有打出来，他是有全都打，
真的假的？
对，然后这样打出来的话，会筛选掉更多，就是会有一些就是你单纯打这个基因就有 paper 出来，但是再加上这个癌症的话就没有。
看，让我重新筛了。
嗯，
哦，头好
你可以
痛，
再自己试看看。只是他的意思是，哦，吓死我！他的
什
意思是
么声音？
这样吗？那个剩10分钟，
哦啊，不知道。可是看一下 breast cancer TH，这样哎，它怎么就没有 THSD4？
有2笔。
哪里有 THSD4？没有啊
嗯
，THSD，
嗯，
哎，有5个那个，哎，有哎，
有，所以还是有。
还是有啊，但要完全没有的很难吧？ 都有啊。
我那时候是有找到、
随便找一个
不然你找，我那时候是找 PHGR1，看一下他们。对啊，像我这样查，它就没资料。你看哦，等一下，这有画面吗？对了，我看我那时候这样查，像这个基因，我就这样打，这样子就没有资料。然后如果你单纯这样打的话，它就有资料。那这个资料可能是做其他的做其他的研究的，只是有提到这个基因。我不知道他的意思会不会是这样，你有没有筛选好？像这个可能就是在讲淋巴结。所以那我的话，我就是一样，我当初标出来绿色的，然后再去什么？我快忘记了，是用
就是去找他的 family 啊。
那什么
找他的
family member， 这翻成中文是什么意思啊？刚刚一直在想。
是吧？有连结的吗？反正就相关的基因啊，那个基因相关的基因。对啊。
好，然后礼拜日要再密测一次嘛，对不对？
头痛了。
礼拜日现在礼拜2嘞，那我们自己要再做出来。好喝。
叫他找时间做吧。
可以啊，我
讨厌。
明后天要去花莲呐。
真的跟你们家吗？
对。所以你现在在屏东吗？
没错，我在原子姐的哥哥家。
哇，哥哥家。他怎么不是在他家？
可能不想让我见到人吧。因为他哥刚好出门，所以请他帮忙顾狗，所以他家现在没有人。圆圆，看一下狗在不在。圆圆过来。
我看一下，
过来，
我家的那只笨猫嘞。
过来，过来。耶！
我就抓我家那只笨猫，好大只柴犬哎！它很重嘛，狗感觉都很重。
它其实蛮小的，它比我家那两只都还要小。
你家那两只，你知道高雄的？
我可能拿远一点。对啊。
好可爱，好可爱。它都不会叫，好乖哦。
对啊，可是它可能我不知道，它被一些奇怪的东西触发就开始大叫。
它比我家好多，没点点嘞。
亲人的。
再问一下。合适啊。我家的猫只要一抓就开始鸡叫。
它怎么样叫？
然后我把它抓过来。它在这里。点。一抱就开始叫。
好大声！
在那叽叽叫。这样，开始叽叽叫，然后那个脸就很臭。
好可爱。
剩下他了。
哎，他有点，哇，好难过。
对啊，
他过世的时候，他没有很难过啊，他没有忧郁。
妈，他过世那个琥珀走了，他有没有？琥珀走了，他有变难过吗？我看起来没有。有啊，还有屋主哦。啊，你踩我电脑。哦，真的，还有屋主哦。真的可能是因为有外人来，还是他有感觉出来？我现在感觉不出来，琥珀走了，他有什么？
没感觉。
我觉得是陌生人吧，而且又有不同的味道，然后那个味道一定是他不喜欢的味道啊，又有那种药水啊什么的，那是对啊，那种麻醉药味道，对啊，那几天是那他现在就很正常。啊，百度娃娃娃，对啊，还要再找时间带他去看医生，这一次也要健康检查一下。
你刚刚吃什么？你刚刚
我说这一次也要带他去健康检查。
知道哦？你刚才说，你不是说你是吃到忘记要开？
就吃晚餐，然后吃一餐，哎，对，干，而且也45了，赶快去联络一下他。
在家里面吃。
对啊，我们在家吃，
哦，
然后把机车记下来，对啊。哎，对啊，好，我先这样，我再跟你用讯息聊。
好，
好，拜拜，拜拜，拜拜。哦。